Good afternoon, good morning, good evening to everyone who is here. I'm delighted to be here today to share part of the work that I've been working on, especially this. I'll focus on the phytoliths research in larger East Africa. I'll be presenting about phytolith research in East Africa, and I'll highlight major milestones and opportunities and challenges that we face in East Africa with these National Museums of Kenya, the Department of Earth Sciences, and working in palynology and paleobotany section. So I'll start with giving, because I'm sure most of us may not be very conversant with what phytoliths are and why they are used and where and when. So I'll go ahead and define some of the terms that those people who are working in phytoliths will commonly use. Phytoliths are opaline silica bodies formed within and around plant cells. They are formed when the plant takes in silicic acid in water, and then the silica get deposited within or between cells. So once the transpiration happen in the upper structures of the plant, the silica get deposited within the cells, and so they take the shape of the cells, forming cast-like. So they are kind of duplicates of the plant cells. So people who have studied plants, especially physiology, they'll be able to tell where these phytoliths are, or the silica bodies are formed. And so different plants, because of their transpiration, the environment they are growing in terms of precipitation, in terms of temperature, the rate of deposition is different, and the alignment of the cells are different. So the most important thing is to understand how the physiological process of the plants are, so that you can be able to interpret the silica cells that are eventually produced by the plants. Mostly they are deposited in the stems, seeds, fruits, and the barks of the wood and roots, depending on the taxa that is depositing them. Mostly the monocots are the most popular in silica deposition and subsequent production, especially the grasses. And we have quite a number that are quite diagnostic. We can be able to differentiate grasses at different subfamilies level, which is a very nice thing and a very crucial opportunity for paleoecologists, because they are able to reconstruct the grasslands dynamics, which otherwise you wouldn't be able to reconstruct using pollen analysis, because pollen across the grass family is similar, very difficult to differentiate between grasses. After the plant dies and decomposes, the silica bodies, these silica cells are reverted into the soils, and they can preserve for a long period of time. And they have a good advantage, especially in the basins where oxidation takes place. And organic matter is not well preserved. They are a good, I wouldn't say alternative, but they are good resources, proxies to use for reconstruction of the vegetation in the past. There's always a problem in trying to change my, okay. I'm going to show you a few common morph types that are used commonly. On my left side, the grass morph types, the first column mainly biolobates, and they are very helpful in differentiating the C4 grasslands, especially the tall panicoid grasses versus short crolidoid grasses. We have crosses and we have sandals that are very diagnostic in chloridoid family. Then on my right, we have other morphotypes that are very, they are good indicators of woody morphotypes. The woody vegetation, some of them are very unique, especially like the one which is ecnate. I'm pointing with my pointer, the ecnate. These ones are directly from the palms. And as I said earlier on, monocots are very good in producing phytoliths and diagnostic types, and palms is one of the monocots, and grasses are also part of the monocots. That's why they give us a good opportunity to be able to understand the past vegetation. I'm going to mention something about palms, because they, especially in prehistoric basin environments, they are indicators of high water table or freshwater resources. And these are nice, this is a good information to understand the environment that past animals, past humans were interacting with, and how the environment was across the landscape, other than looking at the temporal scale, but also spatially. You can be able to tell whether we have heterogeneous kind of vegetation cover, or we had some water resources within maybe a dry basin and things like that. So they are quite informative. Excuse my soul. Examples, or more examples of phytoliths. We also have a lot of noise in the phytolith assemblage. They tell us, we don't ignore them, because they tell us the presence of certain plants or certain groups of plants, although they are not like quite informative in letting us understand the exact species that were growing, or the exact taxons that were growing on the landscape. The other key, as I had mentioned earlier, the palm phytoliths are very informative in telling us about the wetlands, freshwater resources, high water table. And the bottom ones, I'll show with my cursor, they are the conical shape. They are from sages, and they are also indicating wetlands within the environment. So a big question maybe people would ask, with the challenges of the noise, why do you have to study phytoliths? Because I'm sure people who have studied phytoliths here, they can tell you it's a bit challenging, but eventually it pays off when you work with them. So why do we study phytoliths? First of all, it's because of their inorganic nature. They preserve well and long in most deposition regimes, irrespective whether it's very dry or very wet. At least you can be able to get sufficient amount of phytoliths. So they are popular in our basins, and especially like in East African region. Most of the vegetation environmental reconstructions we do trying to explain how the human evolved and what drove the evolution of human and other mammalian. And mostly it's because of the environment they were living in, or the climatic change and things like that. And for us to be able to tell apart, whether it's tectonic, climate, we have to look at the how was the environment? How was the environment changing? Did the vegetation change due to climate change or due to tectonic processes? So for us to be able to understand all this, we need to understand the kind of vegetation that they lived in and interacted with. And for to be able now to do that, that's why we study plants remains. And mostly the plants remains in the dry basins. Quite often we fail to get the pollen or organic material, and phytoliths are coming in handy. The other aspect is they're microscopic in nature, just like most of the plants microfossils. So within a very small volume of sediment, we can be able to extract enough phytoliths assemblage that can be able to tell us the story of the vegetation composition. And then extraction in the lab, we are able to separate them from the silica, the other silica content that is to reduce the noise and during the microscopic analysis. So they have a specific gravity between 1.5 and 2.3, which makes it favorable to separate them from the sandy particles. Specific morph types are diagnostic to plant taxa, a family to species, and this one it's more advantage. It gives a more advantage to the grasses. As we know, the global vegetation cover, we have higher proportion of grasslands. So unless you understand whether you are dealing with the tall grasses or short grasses in the arid regions, it's very difficult to be able to tell what kind of environment you are talking about or has evolved over time. So phytoliths have given us this opportunity to be able to tell apart the kind of grasslands we are talking about, and even further, to tell us about the altitude and the climate in terms of temperature. If there are more poetic grasses, then we know these are high altitude grasses. If we have more panicoid, then we are dealing with a situation where the environment was humid and was warm. We have chloridoid short grasses. These are C4 grasses that are very common in our savanna grasslands, and they are short grasses. Here, I'll give a little bit of historical background when the phytolith research started. Compared to other proxies like pollen, phytolith is relatively younger. And it was recognized and studied from a dust that gathered on Darwin Beagle voyage in early 19th century. And it was defined as plant stones, of which this is what they are. But initially, they could not tell whether it was silica stones or what kind of stones they were, but they were referred as plant stones. And since then, they have gradually gained popularity, especially in archaeological situation where to reconstruct early domestication of plants and paleo diets. Things like corn, cereals are grasses, and they are very significant in the human life since the historical record. So people have use the diet, the cereal diet for quite a long time, but we are not sure from one continent to another when the corn started, when the rice farming started, or things like that. So phytoliths are good in trying to solve these questions. We have advanced into reconstructing vegetation history through time, and especially in semi-arid regions where mostly pollen grains is proving to be quite difficult for preservation. And especially from my experience, I've worked in two basins, Turkana Basin or Gesaila Basin, where the proportion of pollen preservation and plants organic material is very minimal and very insufficient. And when you look at the phytoliths, it's quite well preserved and quite informative. And then recently in 2008, coming forward, the phytolith researchers have developed phytolith indices that are climate proxies. For instance, climate index, aridity index. These are helping us to understand the kind of climate the assemblage one has is representing. For instance, climate index is where you look at the proportion of poetic grasses in relation to the C4 grasses, the panicoid and chloridoid. Then aridity index is the proportion of chloridoid grasses versus panicoid grasses. When you have more chloridoid grasses in your assemblage, then it's a more aridity arid situation, as opposed to when you have more panicoid grasses. Then we have there's a tree cover density index that is telling us about vegetation structure. Are we dealing with open grasslands? Are we dealing with wooded vegetation cover? So when we have a lot of grasses within the assemblage, then it is telling us, because there are specific types that are used to to compute this this index. So you'll be able to tell whether you are dealing with a wooded vegetation cover or open grassland cover. In East Africa, phytoliths research generally initially has been led by mostly by non-African scholars, and the discipline is relatively new. So even in the higher learning institution, it has not really gained a lot of roots. So most of the those who have trained in phytoliths have trained abroad. In sub-Saharan Africa, the main key players, I may not have all of them, but I'm just going to quote the main who have worked in different parts of the continent, African continent. Alexandre from France and their team, they've worked in North and Central Africa. Then we have Doris Barboni from also France, who has worked in North Africa, South Africa, Central Africa, and East Africa. Brembon has worked from France as well, has worked in North, Central, and East Africa. Then we have Frank Newman, Germany, who has worked in North and Central Africa. Julio Macada is Spanish, but in Canada, has worked in Central and East Africa. Marion Bamford, who is my immediate supervisor from South Africa, has worked in South Africa and East Africa. Then we have Julius Leju, he's Ugandan now, is one of the pioneer now from the East African region, and maybe actually the Black African, is from East Africa. He's worked in banana phytoliths from some of the sites in Uganda. Then Alice Novello from France, she's worked with in North, South, Central, and East Africa. Irene Esteban, she's Spanish, working in South Africa. Myself from Kenya, working in East Africa. May Muruji from Uganda, working in South Africa. Then recently we have Chad Yost from USA and also working in East Africa. So far, you can tell from that small cluster how the situation on in terms of the involvement of Africans in the research of phytolith in the East African region, or at large in the African continent. There are a few maybe I could forget, but those are the main that I'm just giving an example. Then at least we are hopeful because we have people still who are interested working with the phytoliths, as challenging as they are at some point. We have Dr. Macarius, who recently graduated from University of Calgary in Canada. He's Tanzanian. Then I'm working with Miss Mariam Budala, Miss Husna from Tanzania. Both are from Tanzania. Budala, she's a PhD student from the University of Calgary, and Husna is a master student from Nairobi University. And then we have Munyaka, who is in our lab and also pursuing to see if he could get registered for a postgraduate. So in a nutshell, that's how we are at the moment. So I'll give you the major milestones that we have achieved so far by working with different research groups. And mostly you realize as I go on, most of these projects are involving the basins, prehistoric basins, where anthropologists, paleoecologists, they want to understand what kind of environment the hominids, the hominoids, and all these ecosystem, how the interaction was between species. And for vegetation is one of the key and the primary characteristic to be able to understand exactly what was going on. So one of the major project, which is research on East African cattle line and hominoid evolution, that has involved two East African countries, Uganda and Tanzania, and it is a very multi-proxy, and people from different disciplines have come together to answer the big question on how the East African cattle line and hominoid evolved during the Miocene. So we're looking back to 22 million years back. And the list of the people listed there the main people that we worked with. And the main breakthrough about with the phytoliths in this research is the fact that we were able to see C4 grasslands. So because the C4 grasslands are believed not to expand, they were thought to expand after Miocene and not to even appear during the Oligocene period. But now we are looking at the early Miocene sites, and we are seeing signal of C4 grasses. They are not many, but at least we have them. We have chloridoid, we have panicoid. So the map on my left is showing different sites where the project worked on. Buluk in Kenya, Western Turkana, Bukwa, which is at the border between Kenya and Uganda, but is accessed through Ugandan side. Moroto and Napak are both Ugandan side. Then we have Tideret, Rusinga, and Karungu, both all from Kenya. And looking at the side, my right side is how these sites are related in terms of what findings have been found there. So that's why they are fossil localities, and the team needed to understand because of the different fossil fauna assemblage, what is separating them. Within a similar time frame, you find the assemblage of the fauna assemblage is different. So the key question which phytoliths and vegetation reconstruction is supposed to address is how are they varied in terms of vegetation? How are these environment varied? There are people who are working with the stable isotope analysis. Others are looking at other microfaunal assemblages to look at different paleo-environmental signals. So we have different groups and even geologists to look at the impact of tectonic processes into all these feet. So initially we were actually, what was interesting about these sites, because it's very sensitive, we are dealing with a time where it could raise some question mark, whether for sure the C4 grasses were there. So I was working with Alice Novello, but she was processing similar samples, but from her lab, and I was working from my lab, and then we were comparing notes so that we can be able to tell for sure we are giving the right information. So the preliminary phytolith data from the 6 early Miocene localities from Bukwa, Napak, Moroto, and 3 in Kenya, Kurungu, Tideret, and West Turkana. And these sites are between 21.2 million years to 16 million years. So out of the, let me see if I could change this. Out of these sites, we found phytoliths in Western Turkana, Moroto, and Bukwa, but we never found a lot of grass phytoliths from the rest of the sites, but we are still looking more. We are still searching for more. Nyamazeni. Okay, so Moroto is its date around 21.2 million, and we compared our data set with Alice to see if the signal of phytoliths were still around. So if you look at the top pie chart, okay, we both found diagnostic grass phytoliths. And then on the second part, the Moroto, we also had another set where in Moroto we saw, at least from my end, I could see more grass diagnostic, but from Ali's side we couldn't. But these were true sandals we don't have any conflict to tell, oh, this could be, this could not be. So these were clear morph types that we found. Again, in Bukwa, we did the same thing, and we both found similar grass diagnostic from the Bukwa 19 million years. And from this site, we analyzed 3 samples, which showed the suggested open environments that resemble the modern savanna grassland and with the scarce woody taxa. So this assemblage mostly reflected the local vegetation because we had a lot of sage phytoliths, showing that it was within a basin. So again, looking at how the fossil plants, the higher abundance of C4 grasses, grass morph types, were able to reflect the presence of grasslands, C4 grasslands within the Miocene sites. So on the looking, we also tried to be able to clearly understand what a certain data is telling us. We compared the modern environments, how the phytoliths would cluster. How for instance, in a grassland, how would we see a certain percentage of morph types, so that we can be able to tell, is Bukwa an open grassland environment or not? Is Rusinga an open environment or a closed canopy environment? So we also other than working on the fossil material, we also worked on the modern analogs, so that we can be able to clearly understand the environments and be able to reconstruct them to our best way possible. The other project that is also has been a success is the Olgosaile drilling project. Personally, before working with the Ecology, we had worked, we had analyzed and studied phytoliths from the basin in the outcrops, and the outcrops that are related to certain findings like zebra and extinction of certain animals and large herbivores, and also certain site that they had found a skull of a human or hominin, which is Homo erectus. This team was led by Rick Pott. And then after we found that because for a long time we had tried to process for pollen, but pollen was quite minimal. So phytoliths gave us a breakthrough to see how the vegetation has been in the Olgosaile basin over time. And we realized the vegetation was not one homogeneous kind of vegetation structure across the basin. It was changing just like in most places today. You have riparian environments, you have oasis kind of spots where there is a wetland and things like that. So we were able to seize that signal. So, and this again was a very high focus multidisciplinary project that in one core or in one level, several proxies were studied, and including diatoms, including stable isotope, soil carbonates, and plants, leaf waxes, and all those stuff that were studied. So from the record, it was compared with how from the Paleosol, and to tell us, we computed Phytolith index, tree cover density index, and see how they vary. And from the vegetation, from the Phytolith record alone, we were able to tell, because I want to remind you about the hypothesis about evolution, where different research scientists has proposed different scenarios. And Rick Pott is one of the mover of the evolution. Highly happened when there was a rapid variation in the climate, or variability, high variability in climate. So looking at the Phytolith data alone, you can be able to tell in the very first, between 900 or 1 million to 400, the variation is more gradual. It's not very rapid, as from 400 coming forward to around 200, and especially around 300 something. And this, interestingly, when you compare with the archaeological record, is a time where they happen to have a transition of MSA, Middle Stone Age, from the Acheulean hand axes, which is very popular in, which was very popular in Olgesaile Basin, was transitioned around that time to MSA, Middle Stone Age technology, and it matches where the high variability in climate and environment, environmental parameters, it matches with that transition, which is a very key thing to see and to be happy about. Again, now comparing with the soil carbonate, again, the data still showed the same trend, where after 400, we have high variability time between 500 and 300, 320 especially. We had a very high variability in terms of environment and climate. And this is a time that we see from the fauna assemblage, there's extinction of megafauna, and we have new fauna assemblage in the basin, and again, new technological innovation by the hominin. Coming much later, we worked again with a team that is listed there from a site in Mount Kilimanjaro. This is a highlight site. And the idea was to look at the vegetation change and fire record at high elevation in Kilimanjaro, the last 3,000 years. Phytoliths again came in handy. If you look at the paper, we have pollen data, we have phytolith data, and it tells where we are able to see where the actual change in terms of vegetation within high altitude was, and how this, what is meaning of this, how this relating with the archaeological record within the basin, the lower basin or the lower part of Kilimanjaro, to see whether people were exploiting or they were going there for some resources. So we were able to detect some interaction between the forest and the mountain and the people who are living in the lower elevation of the mountain. So during the Phytolith assemblage, I'll call you to see the upper part of the diagram, the Phytolith diagram, where you see the changes in terms of the vegetation and how, just a second, I see if I could get to the end of this. Okay, so in terms of the trackades and in terms of forest indicators, we can see they were becoming much lesser at the top of the profile, as opposed to the rest, to the other part of the profile. So our main take home message from that paper was the changes between 3,000 to 800 years. Looking at the pollen, phytolith, and charcoal record, it shows a very small variability, especially in the Cretaceous belt, that is the very high altitude in part of the mountain, with apparent multicentennial secular variability and long-term pattern of increasing charcoal. Then from 800 to 600 year, the mountain forest taxa, that is now the data from the pollen, varied rapidly. Cypress increased highly, charcoal became low. Then from 600 to the present, the woody taxa decreased. Ericaceous taxa and poaceae, again, we see them becoming more conspicuous. Then uphill wetland ecosystem are crucial for ecological and socio-economic resilience on the surrounding environment, or the mountain is. Then the results were, these results, vegetation results, we correlated this with archaeological data. One of the co-author is has done some work from the Amboseli Basin, some sites that are related to Kilimanjaro, and we were able to understand the complexity of Kilimanjaro and how the historical human interacted with this environment. So these paleoenvironmental records create a long-term context for current climate, biodiversity, and land use changes on and around Kilimanjaro. Again, also it raises questions, what is happening in other mountains, and it's very important that people can think about how other mountains, or how people related to with the other mountains in the highlands. So the next presentation, phase is the opportunities and the ongoing research projects on phytoliths. The first that is very important is to integrate, to have integrated calibration of modern climate and vegetation proxies in Kenya. It's a project where we are collecting, or rather we already collected, not from all sites that we thought we could, but we have already started collecting data sets of different proxies like phytoliths, biomarkers, pollen, soil carbon, and the main goal is to improve our ability to reconstruct the past environments so that we can see how clear or how close, how accurate will the fossil data be able to tell, to reflect the environment in the past. So to see in terms of percentage, is it 99% accurate, or is it 50% accurate? And looking at different proxies at the same time from the same site, how are we adding value to that? So this is a project, again, that is in collaboration with various people from abroad and from who are local also, and it's still in process. So far we've collected samples from Mount Kenya, Kakamega Forest, Lake Nakuru National Park, Samburu National Park, Meru National Park, and Nairobi National Park. If you look at those sites, they have, in modern times, in present day, the contemporary vegetation is differs from one site to the other. So when we are speaking of high altitude vegetation, Mount Kenya is there to represent what is the accurate proportion of phytoliths or other proxies that can be able to tell us, oh, this was like this or like that. In Kakamega Forest is also a unique forest. It's not a mountainous forest, but it is more a low altitude forest, but very rich. And mainly it compares with the Cameroon forests, or maybe Congo forest. Then Lake Nakuru, these are basically in the savanna, woodland, and bushlands. The Samburu National Park is more dry. Again, we have Meru National Park. It has its uniqueness in terms of the relationship between precipitation from the Mount Kenya. It's really affected in terms of precipitation by the location of Mount Kenya. And Nairobi National Park is also a highland, and which has a lot of impact with the human in the city. And the studies, the proxies that we're looking at, studying are the pollen, phytoliths, leaf wax, soil carbonate, and the modern vegetation. So we have a team that are representing all that. The other project is looking at the fire history. And in East Africa, in Kenya especially, the fire has been at least recognized in East Turkana Basin, Kobifora region. And we want to look at the role of fire in the changing hominid behavior, because we know their eating habits, the diets maybe made them become stronger, wiser in terms of their brain and thinking. So the role of fire in the changing of human behavior is critical for those who are studying anthropology. So my major research role in this one is to look at the phytoliths, how the vegetation was, that is the main thing. And then from those fire hazards that we already identified in the site, to look at the burnt phytoliths. If we can get them, then we can be able to tell whether for sure the fire was there. And now we can discuss from there what was the influence, how did the fire influence the humans' behavior. And this the site dates back to 1.8 to 1.6 million in early Pleistocene. So as I say, the project seeks to evaluate the hypothesis that the controlled use of fire became an important element of hominin adaptation at Kobifora after 1.6 million. And then the main thing we are looking at is the biomass burning, which includes the plant waxes. Then we have to look at the microscopic traces of burning. FTIR, this is a method where they're able to detect the burning, how the burning was, whether it is controlled or deliberate, something like that. Then burnt phytoliths and charcoal. So charcoal, at least most of us who understand about charcoal, the local charcoal from the local deposit will show you, will be larger in terms of size. So you can be able to tell between local fire from the charcoal or the regional input. The other one is also a collaborative project, also looking at the Miocene. It's actually like a continuation of the previous Vichy Miocene project, but this one is based on, you need to apply high resolution basin forecast approach where different people, geologists, who want to look at tectonics, volcanism, erosion, climate, in a nutshell, is to bring the earth, life, and nature and climate science together to be able to solve what happened through to Miocene, because this is where mainly the hominoids, the gorillas or the monkeys and all that were evolving. So what happened? What kind of vegetation did they live in? What kind of tectonic processes happened around that time, and what was the impact of this to the life that was there at that time? And then these are recent project that we are working from Aberdare. It's basically to look at the historical ecology and to use pollen, phytoliths, and plants and archaeological record to be able to tell the impact of human before colonial time, during colonial time, and after colonial time. These sites are already documented historically that they were occupied before colonial time. And then there was when the colonial masters arrived in Kenya, the way they came to the highlands and it located people and it settled them. Some of them were settled in the forest as forest scouts. And there were villages within the forest that have been there since colonial time, and the people were evicted in late or mid 80s. And the idea is to look at that impact. How did they impact the forest during colonial time and before colonial time, and what is happening now after being evicted in the forest? This is an idea born because of what is happening, especially in Kenya. Mostly we have issues, conflicts between people who are living in the forest and the government, and for them to be able to tell whether humans are literally, because the argument from the science and from the local community and from the government is completely different. The local community feel that they've been in this forest, they have lived in this forest for many years, they cannot even tell for how long, and they feel like they have never had like negative impact on this forest. So they feel like it's always political when they are being evicted. But so we want to bring the science aspect to see whether for factual, whether the government is right or the people are right in terms of how people relate to, especially people who have been indigenously living within the forest. So this is again an ongoing project that we just finished field work, and we are about to start the analysis and see how they relate. The challenges in East Africa, in Kenya, is limited funding. It's very difficult to get funding, especially from the government for basic science. So they would always get biased to looking at to funding innovative sciences or applied sciences where the agricultural impact, like where there's a like immediate return to the community. But when they look, the kind of science we do, they feel like it's too basic for them to, it's never prioritized. So mostly we have to collaborate from people outside the country, and also to seek for funding from funders, from organizations that have similar interests. Limited infrastructure, like for instance, the lab, palynology lab in East Africa. I think we in Kenya, we are the one heading the three countries, the Tanzania and Uganda, in terms of facilities and all that. And we cannot say that we are fully equipped, like you'd imagine in other countries, but now that's the situation it is. Then in the society today, the perception about basic science, it doesn't pay immediately. So many people want to study things like economic, they want to study things that are paying back soon, or things that, courses that will get them jobs that are business oriented, or jobs that will give back a lot. Definitely you will all agree with me about science and payment. It's not the best, but again, the satisfaction about working and research in this situation, it depends with especially the scientists. So that's another challenge that we have. And looking at the basic science in general, and narrowing down to looking at some, like you have to spend hours and many hours on the microscope to make sense of what you're doing. Again, not many people are patient with that, especially students, they are not. So these are a few people whom I'm going to acknowledge, especially National Museums of Kenya, which is my parent institution, and all these other institutions, because naming individuals would take forever. It's a long list, but that's why I decided to name the institutions that we have collaborated with, among others. And publications that are like directly from the work we do. This is what I have on the screen. Yeah, you can have time and check. I think I'm done. Thank you so much. I hope I didn't get lost somewhere. And the noise in the background, I apologize. Good, thank you.